Good afternoon, everyone. I'm Nárai from the Brain Imaging Center Research Center for Natural Sciences, where I perform eye movement research under the supervision of Dr. Béla Weiss. Today, I'm going to present our paper titled Predicting Reading Speed from Eye Movement Measures. Reading has a pivotal role in our society. However, about 5 to 10% of the population experience reading difficulties. With the long-term goal of investigating dyslexics, we are trying to better understand the reading process. It is known to be sensitive to low-level visual features such as letter spacing or font type, linguistic characteristics like word frequency and word length, and high-level cognitive processes like attention. These effects are reflected in eye movement events, such as fixation, saccades, and corrective eye movements, which are called glissades. Choosing reading speed as a measure of reading competence, we asked the question of how eye movement measures affect reading speed. To this day, only a few studies investigated the effect of eye movement measures on reading speed. The 3 studies listed here investigated children, adolescents, and typical and poor reading adults. Although they investigated different populations and also used different methods, they reported similar results. For example, fixation duration negatively impacts reading speed, meaning that participants with longer fixations read slower. The same is true for the number of forward and backward saccades, meaning that with more saccades, reading becomes slower. Finally, they reported increasing reading speed with increasing saccade amplitude. Despite these results, to the best of our knowledge, no study investigated the relationship between reading speed and eye movement measures systematically, and also no study investigated the effect of glissades. To do this, we used the eye tracking data from one of our natural reading experiments, which was conducted with the participation of 24 young adults. Eye movement data was recorded using an SMI iView eye tracker, and we also recorded EEG, but it is not used here. The participants were instructed to silently read 32 Hungarian paragraphs in their own natural pacing, like as they would read the news. The paragraphs were presented line by line in 3-letter spacing condition, as you can see on the figure below. However, for the current analysis, we only use the data from the normal spacing condition, which is presented in the middle. We also presented test statements for comprehension, where participants had to report whether the statement was true or false. Using the eye tracking data of this experiment, eye movement events such as fixations, saccades, and glissades were detected using a velocity-based adaptive algorithm. Based on the features of these eye movement events and based on text properties, we created 23 global eye movement measures, which were estimated on the subject level. We have median measures such as median fixation duration, also percentage values like percentage of forward saccades, and frequency measures like frequency of forward saccades. We also calculated the subject level reading speed based on the same experiment. You can see here the correlation matrix for all measures, and in the blue rectangle, the significant Spearman correlation coefficients between reading speed and eye movement measures. Several eye movement measures positively correlate with reading speed. The strongest ones are median forward saccade amplitude and median saccade peak velocity, while there are measures negatively correlating with reading speed, such as median number of forward saccades normalized to word number. However, inspecting this correlation matrix, we see strong intercorrelation between several eye movement measures, which imply high degree of multicollinearity in our data. Therefore, the use of correlation analysis or simple linear regression was not sufficient for this study. So we use linear regression with ElasticNet regularization using the ElasticNet CV class from the scikit-learn package. We applied 10-fold cross-validation, 20 L1 ratio, and 20 alpha values. We used reading speed as a dependent variable and 4 subsets of the 23 global eye movement measures as independent variables. I will talk more about these subsets later. And we also z-scored these variables, so the models will be comparable between each other. We use predicted R-squared as performance measure, which was calculated as the median of R-squared values from a 10-fold cross-validation. And finally, the significance testing of regression weights was performed using a permutation approach with 1000 random permutations. Here you can see the regression weights for the model using all 23 eye movement measures marked by a blue rectangle. The significant coefficients are marked with black rectangles. This model covered almost 90% of the variance in reading speed, and we found 3 significant regression weights. Median forward saccade amplitude and the frequency of forward saccades positively influence reading speed. Thus, subjects with larger forward saccade amplitude or higher forward saccade frequency can read faster. However, median number of forward saccades, which is normalized to word number, has a significant negative weight, thus predicting a decrease in reading speed. Since the frequency and number measures of saccades are closely and also intuitively related to reading speed, they may mask the effect of other eye movement measures. To investigate this, we excluded the frequency measures first. We excluded frequency measures from our next model, which covered about 50% of the variance in the reading speed data. While both the amplitude and the number of forward saccades remain significant, we have 2 new significant measures. Increased median fixation duration predicts slower reading speed, while an increase in the percentage of forward saccades predicts faster reading. These results are in line with the literature, except for percentage of forward saccades, which was not investigated before. And we also found a marginal effect of median backward glissade duration here. Next, we trained the model leaving out only the saccade number measures. This model explained about 80% of the variance in reading speed. As you can see, the weight for median forward saccade amplitude became much stronger, and the percentage of forward saccades became significant. This is probably due to the strong negative correlation present between forward saccade amplitude and the number of forward saccades. And finally, we trained the model dropping both frequency and number measures of saccades, covering 44% of the variance in reading speed. Here you can see the combination of the effects observed in the previous 2 models, which are noted here as set 2 and set 3. So, as a conclusion, primary 3 measures of forward saccade drive reading speed. These are namely amplitude, frequency, and number per word of forward saccades. Saccade frequency measures included here may mask the otherwise significant effect of fixation duration and percentage of forward saccades. And normalized saccade number measures reduce the effect of saccade amplitude. And finally, we found the potential importance of glissade measures. However, further investigations are needed to reveal the exact effect of glissades in reading process. In our further work, we plan to investigate the effects of letter spacing modulation on regression weights and try to answer a question of how does the regression model depend on reading abilities. This study was supported by the grants listed here. And thank you for your attention. If there are any remarks, suggestions, or questions, we are happy to answer them online, but we can also be contacted via the provided email address.